really interested in learning much more about the domestication of various finches. So one of the reasons I've come along today, and I'm very I thank Sam and Don for getting in touch and coming and visiting us in the group.
Sorry, sorry, can you leave them on because we're videoing?
Oh, Chris, sorry. Turn them off.
Sorry.
So you're the so I'll turn them on.
Sorry, Simon.
That's all right. So yeah, so I'm basically here because I want to start. We want to start engaging and learning more from you guys. So the work that my history is that I started off working on birds nearly 20 years ago, actually now in the UK. Then moved around to Sweden and Oxford, working on a variety of birds in the wild. And then I moved to Australia just over 10 years ago now, and I work on a range of birds, so I lead a group at Macquarie University. We work on a range of birds. But the favourite work that I've enjoyed doing over that time and the most productive work is on a number of finch species. And I'm going to talk to you a little bit about some of that work tonight. But we also do other work. We're working on wild birds in the desert in far west New South Wales, where we're studying questions like the evolution of cooperation. This is a babbler and the apostle bird. And these are essentially behaving like meerkats. And there's a lot of very interesting questions about those. But the three finch species that we've been working on are the zebra finch, Gouldian finch and Longtail finch. All of this work's been going on for almost, between 6 and 10 years. And all of that work is done from both in the wild and also in the laboratory. And that enables us to understand the birds in their natural habitat and try and understand how they've evolved in the ecology, how they fit into the habitat and the community of other birds. And we do that by catching them obviously and sampling them. We've put nest boxes up for all of these species in the wild, which means we can get many hundreds of pairs each year to actually focus on. And nest boxes are really good because the birds essentially are coming to us and we get very easy access. We can catch the adults, we can bleed and sample and look at do experiments on the offspring as well. So the main field sites, we have a long-term site up in Wyndham in WA, where we're working on Gouldians and Longtails. The main zebra finch site is near Broken Hill, but we're also starting work this year at Alice Springs and hopefully up near Mount Isa as well. And then the Longtail finch, we've actually been working, we've done a big transect where we've caught birds about every 100 kilometres right across the whole of their range. Most of the questions that we're looking at are really about the species. And when we think about animals, as I'm sure you're aware, we kind of think we know what species are because we can get the field guide and you can just flick through the field guide and open a page and it'll tell you, how the biological diversity that we see out there is packaged up into species. But one of the things that biologists spend a lot of time thinking about is what actually is a species and how the species evolve. And of course this is something that people have been thinking about a lot since the work of Charles Darwin. But we actually still are learning an awful lot year by year about these questions still. So essentially species, from a biologist's point of view, is that biological variation is packaged into species, and these species aren't actually as distinct as we think, but the way we define them is it's the most inclusive set of potentially interbreeding individuals. And by interbreeding we mean that they breed together successfully and that their offspring are viable. So hybridizing, which of course some of you will be familiar with, that's not really, you can hybridize between different species, but the offspring aren't always viable themselves. A good example of the speciation process that people have been working on and we're working on is the Longtail finch, which has 2 subspecies, and some genetics that was done at Harvard University in the US has pretty much shown that these two shared a common ancestor about 300,000 years ago. And then they shared a common ancestor with the black-throated finch, which is seen as a separate species from these two, about 600,000 years ago. And then the ancestor, of course, we don't know exactly what the ancestor looked like, but essentially the ancestor to these, those two, which are recognised as kind of subspecies, so things start becoming a little fuzzier. There was an ancestor that presumably looked something like all of those. Now, the way we think these particular species has evolved is that probably 1.5 million years ago, the top end of Australia was all suitable habitat for the ancestral species. So they might have occurred across the whole range, right from the Kimberley, right across to northern Queensland. But then we know from lots of records that the climate about 500,000 years ago was extremely dry. The deserts pushed the suitable habitat further north, so that the top end of Australia, as far as a grass finch was concerned, was restricted to the 3 northernmost tips of the continent. So then possibly for 500,000 years, the ancestral species was isolated into 3 little discrete units, and within those units, over time, they just accumulated mutations and changes, so that when the continent became relatively wetter again, and the habitat, so this is the savanna habitat that these grass finches live in, as the habitat came further south, those 3 refugial populations came back into contact with one another, but because those different populations had accumulated all these changes over time, when they came back into contact with one another, they couldn't interbreed. So they'd become different species because when they tried to breed together, they just didn't succeed. And that's why today we find yellow longtails here, red longtails here, and yellow-billed ones here, and black-throated here. It's because they're unable to successfully interbreed across those points of contact. So what those points of contact tell us, if we can actually ask questions about this, what is it that's stopping them interbreeding? Is it behaviour? Is it their genetics? And that's what we're able to do with these finches, which are really good model systems. So again, just to remind you again that selection that we're talking about here, this is what Charles Darwin was focused on when he was publishing his very important books in the 1850s and 60s. And there's really 3 forms of selection. Natural selection, which is the survival of the fittest, that you're probably familiar with. That's about things like, cheetahs running or gazelles running faster than cheetahs and getting away. So natural selection is about surviving, but it's not good enough just to survive. You need to reproduce as well, because what's important is that you leave ancestors behind. That's sexual selection. So it's the variation that you get across a population in how many offspring different individuals produce. And this sexual selection acts on things like ornamentation. So the colour that we're all interested in our birds, that colour has evolved through the process of sexual selection. The most attractive birds produce the most offspring. And then the other thing that we're becoming increasingly interested in is artificial selection. And Darwin did a lot of work on this. He did a lot of work on actually pigeons, on the domesticated forms of pigeons. And it's actually really amazing that just last year, so whilst Darwin wrote a whole book on pigeons and the different forms that the pigeons had taken, purely through artificial selection by pigeon fanciers, a paper that was published last year in Science, which is the most important journal that scientists have for publishing their work, actually was the first paper that's been able to look at these. This is just 4 examples of the different strains of pigeon that are out there, and they're all connected by these weird crests around their head. And this team, which was a team of about 25 scientists in the US, they actually managed to they identified the gene which mutated several hundred years ago, and a single ancestor has given rise to all of these different strains. And they've actually unravelled that. They've identified the exact mutation that's caused that, and it's now common. So that one bird that had that mutation has now, that gene is now common through all of these different forms. And then this is actually, if you like, it's a representation of the family tree of all of those different ornamental strains of pigeons, and all of the ones that have the crest are related to each other. So this is a really kind of cutting edge paper, and interestingly enough, it's focused on this whole artificial selection that Darwin himself was focused on. So before I moved to Australia, I was very interested in these kind of processes, and this is a blue tit, which is a very common passerine in Europe and well, across northern Europe. And here in Oxford, we had a population of about 300 to 400 pairs each year, and this is just an example of it. So we were interested in selection on colour. So we measured colour with a machine that measures it very accurately. And we could measure the different colour patches, and then we actually looked at how that selection acted in the landscape at quite a small level. So this woodland is only about 7 kilometres from here to here, but there were patches of the woodland that are quite low down, low lying with a bit of dampness, and then there's a patch up the top which is quite high on the hill, and the soil's different and the trees are different, and within that woodland there's very strong selection. So the colours here represent the type of selection essentially on the blue head. So all of the birds down here have very blue heads, whereas the birds up here, their heads aren't as blue. So you can measure this selection at a very fine scale, and this tells us something about the fitness, or how many offspring an individual will produce, depending on the head colour and where it happens to live at quite a small scale. And when I moved to Australia, I wanted to look at these exactly the same questions, but over a much larger scale. So again, this is the Longtail finch, and we've got the same kind of thing. We essentially have variation in colour across the whole range of the finch, and the colour that you have will affect how fit you are depending on where you are in that landscape. The great thing about the Longtail finch, as I'll come to later, is that of course you can take birds from here and here, and you can breed them readily in the lab, because I mean you guys have been doing that for years. But you can't do that with many of those European birds that people study. And of course, the things that we're learning about with finches apply to most birds. So if you actually go back to the bird guide, even though we kind of look and we think we know what a species is, actually most Australian species have quite a lot of this subtle variation in colour. So here's just 3 examples which you're probably all aware of, where there's subtle, or sometimes very discrete variation, so red and yellow faces. With the pardalote, there's very minor details, like the number of spots they have on the wings. And then there's crimson rosella, where there's a kind of a very strange geographic pattern of colour. But it's the same thing, and essentially this is what's driving, this is the engine that drives the diversity of species. It's breeding with one another, because that's the formation of the species. And we've been doing that primarily in the longtails, where there's just 2 forms, and the Gouldian finch, where there's 3 different forms. And the genetics underlying these different colours are actually quite different. So in the Gouldian finch, those 3 head colours are very discrete, and again, you guys would know all this. So that if you breed a red and a black bird, you don't get kind of a half red black. You either get red offspring or black offspring. So it's what we define as a completely discrete trait. The Gouldian finch, if you actually look back so we've done some work on the history of the species. For the first 30 or 40 years after they were discovered, there was a lot of argument actually. There were a lot of scientific papers and discussion about whether it was 2 species or whether it was a single species that was doing some weird stuff. And it turns out that the latter is the case. So there was quite a lot of controversy for at least 30 years about whether it was 2 species. And that controversy was actually only resolved by some essentially finch breeding that happened behind the Australian Museum in Sydney here. So the director of the Australian Museum, Ramsey, decided that the only way to resolve this was to take some finches from the wild, bring them into an aviary, and they actually bred Gouldians back in the 1870s, which seems quite amazing. And they did that right here in Sydney, and they concluded, as a result of these results, they concluded that because they had a black pair of birds that produced a red daughter and 2 black sons, they concluded that it must be one species, that there must be interbreeding going on. As it turns out, the Gouldian finch was the first finch, or the first animal, in fact, where people actually described the genetics. So that was properly described scientifically by Southern in 1945. And again, his paper relies on data that he gathered from a group of finch breeders in the UK. And he resolved the exact kind of genetics, the inheritance, which again, a lot of you are familiar with. It turns out that the result they got in the aviary in Australia Museum must have been wrong. A black pair can't produce a red daughter. Okay? So the key thing, the key issue here is how did they get that wrong? They almost certainly, well, they got that wrong because they were doing this experiment, this very early experiment in an aviary. So that red daughter must either have been an egg that was dumped by another pair, or another female, or that female must have cheated on her partner and gone and had a copulation with a red male.
That doesn't happen.
So not in Sydney anyway. So we've gone back and so we've gone and retested those, and we've also looked at the stability of the morph frequency through space and time. So we looked at all the museum records of the populations that were sampled as far back as they started collecting these, and then there's also a number of kind of contemporary historical records. And in the wild, it seems that both across the whole of Northern Australia and also back into time, there's a reasonably stable frequency where in males, 3/4 of the birds are black and about a quarter of them are red. So again, that's something that Darwin himself, not with respect to the Gouldian, but that is a question. How do these things, over time, coexist with one another? Because you'd always expect that if there's two different forms that are genetically distinct, one of them's going to be better than the other, and it's going to outbreed it. And again, some previously finch breeders have said that if you keep a relatively small aviary with reds and blacks, before long you'll just get one or the other. So again, we might expect to see between two separate species, which is a little bit odd. And then we also looked at this whole idea about extra pair copulations. So we did a follow-up experiment in which again we forced females to pair with either a right head male or a wrong head male. And then we took that partner out of the cage for 20 minutes and introduced an extra pair male and gave her an opportunity to copulate with him. And all of the females took that opportunity, which was the first surprise. But what was really striking here is that, so don't worry about all four of the figures, but essentially the important one in here is that when the social partner is wrong, i.e. he's going to be bad news because his genes aren't compatible, when the extra pair male is right for her, nearly 70% of the offspring are fathered by that single copulation. So that guy gets one copulation, he gets 35 copulations while she's laying her eggs, and yet he fathers 70% of the offspring. What that suggests is that there's some really interesting stuff happening inside her reproductive tract. So she's actually essentially filtering out the sperm and biasing in favour of him. And that's exactly what we've predicted. So there's behavioural mechanisms. Obviously females can choose a partner by looking at them. But then there's also these complicated mechanisms that happen inside, whereas they can actually apparently kind of filter sperm or. And again, this is the opposite one. So this is where her partner's a matched one. The extra pair one does get a copulation, but those copulations don't result in any offspring. So females are very clever, both before and after mating. So the work we've done since then, we've been working with people in the UK and in America to actually, so I don't know if you're aware, but in the last 10 years or so there's been a real revolution in our ability to look at the whole genome of an animal. So that means we're actually being able to sequence every single gene, and there are tens of millions of them in each species. And the second species of bird in the world that was done, so the chicken was the first species for which we know the whole sequence of all of its DNA. and the zebra finch was actually the second species that's been sequenced. And because the zebra finch is closely enough related to a Gouldian, my colleagues in Sheffield have actually managed to look at the Gouldian finch, the whole genome, and we've now identified exactly where the head colour gene is. And it's exactly there. And this is the Z chromosome, which is the male sex chromosome. So we now know that the red gene is, the red and black head colour gene is pretty much there. What we need to do next is we actually need to definitively show that that's the gene that's causing feathers to grow either red or black. And so we'd be very keen to get some feathers from some growing feathers as they molt to actually show that that gene is being expressed differently. So that's one of the things that we're doing right now. But the other thing that's come out of this work, so this work's been done on blood samples from wild birds and the domesticated birds. And without really having to worry too much about what's in here, what we've found is that the incompatibility we're seeing in the domesticated birds that Sarah works on with Mike Fiddler doesn't happen at all in the wild. So there's something weird going on there. So what that suggests, so in the wild, red and black birds can breed together apparently without any problems, whereas in captivity, certainly the large population that we put together with Mike Fiddler, those birds were having real problems. So this suggests to us that something has happened in the captive population that's driven very, very strong selection. And we need to kind of think about how the reds and black genes are operating, and the fact that they're actually kind of fighting against each other. So they've spent that time separated, and when they came back into contact, it's as if the genes are actually kind of fighting against each other. And that fight is also played out differently in males and females. And we know that because males and females look very different. So the expression of the genes, how much colour they produce in a male and a female, is very different. And you all know that because hens look different from cocks. But what we've also found is that hens in the wild look very different from hens in captivity. So this is a wild hen, and again we've measured them with this machine which measures the colour very accurately. And in captivity there's two things about hens which are quite different. So the first thing is that they're much more colourful than hens in the wild. And the second thing is that hens in captivity tend to have reasonably clean heads. So a red bird has more or less like 90% of the head is red, whereas in the wild, lots of the red-headed females only have about 5% of the feathers are actually red, and the rest are actually black. So it's almost as if there's a fight happening within each female. They're kind of trying to resist the effect of the red gene. So what we're really interested in doing now, and it would be fantastic if anyone's happy to help out with any of this work in the future. We're actually interested in trying to identify the strength of this artificial selection that's caused this, because it's happened quite quickly. Because probably most of the domesticated birds are still getting genes from the wild populations, and certainly those populations haven't existed for more than about 30 or 40 years. So the selection's been very strong. So we're very interested in trying to identify that, and we're also trying to identify how different females fare when they're breeding with a black male. So does a female that has a quite complete red head fare differently from a bird that has more black in its head? And to do that, it would be excellent if we could actually work with people who are willing to set up some breeding experiments and maybe let us take some blood samples. So that's the end of the Gouldian stuff I was going to talk about. So that's just where we're currently at with the Gouldian thing. Then with the Longtails, again, we're interested in this question about colour and how it varies. And so with the Longtails, what we've done, we've collected wild birds from the Kimberley and also from Queensland, so the two ends of the natural distribution, and we've brought them into the lab, and we're breeding them, or we're crossing them, to try and again look at how that colour is inherited. And again, some of you probably know this. Now the Gouldian finch would be like this, so essentially you get a red and a yellow, or a Gouldian, a red and a black. and you would get offspring that are either red or black. But we don't get that in the longtails. And again, most of you would know this. When you breed a red and a yellow together, all of the offspring are orange. And what that tells us is that it isn't a single gene like it is in the Gouldian. There's lots of genes. There might be hundreds of genes, and each one of those is contributing just a little bit to that colour. And that's why when you breed a red and a yellow, it's the kind of collected, it's basically how many red genes you've got and how many yellow genes, and they're all kind of coming together and fighting, and you end up with orange offspring. So that's the first thing that we've kind of done. And then the second question is, as in the Gouldian, what's the fitness like of these orange birds or these hybrids? So at the moment we're crossing the hybrid offspring, and we're interested in, so again there's a possibility they don't breed very well because of the incompatibility. But the other thing we're looking at is there's genetic, it may well be that when you back cross them with their parents, some of them will produce orange offspring, but some of them might actually potentially throw yellow or red offspring. And again, that will tell us something about how many genes there are and where those genes are. And again, because this is closely related to the zebra finch, there's a group in America that have just they're spending, so they got birds from us, so they got blood samples from us. They've fully genome sequenced this, so this will be probably the 10th bird to be fully sequenced. They've got a team of about a dozen people processing all that data as we speak, so that again we'll have the full genome sequenced for the longtail, and literally within about 6 months. And this is an email I got yesterday from those people, so this is literally, this hasn't even been published, this is hot off the press. And again, don't worry about the details here, but essentially the different colours here are the different chromosomes and different places on the different chromosomes. So again, the whole genetic information is, as you probably know, is split up into different chromosomes. That's essentially the way it's packaged in an animal. And what these people have done is they've compared my yellow bills and my red bills, and they've actually said, where are there differences across the chromosomes? So in some chromosomes, like chromosome number 2, which is one of the largest chromosomes, there's no difference at all between those reds and the yellows. But some chromosomes, there's huge amounts of difference. In fact, so this is the this is the Z chromosome, where all the interesting stuff's happening in the Gouldian. And on the Z chromosome of the yellow and the red longtails is almost completely different. So there's lots of really cool, interesting stuff happening, but just on one chromosome. And it doesn't mean that all of these are the genes that control yellow and red, because those birds are also going to have changed for lots of other reasons, because the habitat's different and for random reasons. And then there's also a few differences on some of these other chromosomes. So these are essentially, this is a way of saying, these are two species that are pretty closely, so in evolutionary terms, they've changed relatively recently, but we can actually now look at all of the genes that have changed and how that's affecting these birds, not just in their colour, but in their ability to breed with each other. I have a student who's working at the moment with us who's looking at sperm in these different forms and how competitive the sperm are with each other. So this is some really cool stuff that's happening with collaborations around the world. And then lastly, I'm just going to talk a little bit now about our zebrafinch work, which isn't quite as complicated and as focused on genetics. And again, we've been working now out near Broken Hill for the last 10 years on the zebrafinch. The reason that we're working on those, they're a very nice bird, but more importantly than that, they're one of the most important kind of model systems that we have in biology. So there's over 1,500 scientific papers that have focused on the zebrafinch, and those papers cover most of modern biology. So there's people who work on genetics, people who work on ecology, lots of work on behaviour, lots of work on neuroscience, so how the brain works, how animals learn about songs, things like that. And Ondi, who's here at the back, she's done her PhD, or finished her PhD in the States, where she worked on physiology and looked at hormones in domesticated zebrafinches in the US. So the zebrafinch is actually hugely studied and literally tens of millions of dollars are going into studying the zebra finch, and yet there's actually very little work done in Australia, and most of the people who have done all that work have never even been to Australia, and they've certainly never seen a wild bird. So one of our big questions is to try and learn about the zebra finch so that those other people understand it a little bit more, because what they're working with are domesticated finches that they literally went to the pet shop and bought, and those finches in America and Europe have been essentially maintained for 200 generations by finch breeders, not by scientists. So again, that's a reason for us to want to understand what's happened in that 200 years. So just a few little things I'm going to tell you about. So the first thing is that bill colour in wild zebra finches is not as different between a male and a female as it is in Europe and probably in North America. So in the UK, if you go and measure them in a couple of populations, male and females have very different bill colours, whereas in Australia, both in captive populations and also in wild populations, they're not very different at all. So that's one obvious difference. And it's easy to see how bill colour might have been the result of selection by finch breeders. Another thing that we found is that wild birds, so if we look at females choosing partners, which is something a lot of people have worked on, wild birds are far more choosy than domesticated birds. But not for things like colour bands or bill colour. So there's been lots and lots of work looking at the choosiness of females over bill colour in Europe and North America, but in Australia females don't care about that. Another really very profound difference is that domesticated birds are more, in Europe and North America are far more promiscuous. So 30% of the offspring, if you go into an aviary, 30% of the offspring in a nest are not fathered by the male who's looking after them at that nest. So that's the rate that's been found in America when people have done the genetics. But in the wild zebrafinch, that basically doesn't happen at all. We find about 1% of offspring are the result of extra pair males, and that's probably where a female has lost a partner and she's repaired fairly quickly. So those are really big differences. And so, as well as kind of highlighting those differences and kind of putting this question mark about where have those differences come from, we've also been focusing a lot of our work on, well, why are zebra finches different here, or what is it about wild zebra finches that makes them so faithful to their partner? So again, there's very little genetic infidelity. We've never recorded divorce in over 2,000 breeding attempts. And more importantly, experienced partners are much better at reproducing, so they can breed together much more quickly. And actually, the other thing you'll see in most of the struded finches is you'll often see birds allopreening with their partner or cozying up to their partner. Now, to North Americans and Europeans, so that probably doesn't seem strange to most of you, because an awful lot of Australian birds do this. You never see a European bird or a North American bird doing that. There's just a big difference, and because I came from Europe and I'd worked with a lot of those other systems, this immediately struck me as something that was a bit kind of weird. But in Australia, it's not at all weird. So zebra finches stick with their partner all the time. So we've been tracking them around the desert with various different techniques, and they're basically always with their partner. They visit the nest together, but they're foraging together. They go off and drink water together. They just stick to each other essentially like glue. And they do that throughout the year, not just when they're breeding, but throughout the year. They're also incredibly well coordinated with each other. So this is, if you measure the amount of time when birds start incubating, so essentially they'll actually start sitting on eggs by increasing the number of minutes per hour. So the day they lay their first egg, a female might sit on her eggs for 10 minutes per hour. Second day, 20 minutes per hour, etc. The amazing thing is that the male and the female both increase the level of incubation side by side. So even on the very first day, when the male doesn't probably really, you wouldn't imagine that he knows that she's even going to lay an egg, he actually manages to coordinate how much incubation he does incredibly tightly with his partner. So this suggests that they're somehow communicating with each other and they're really well kind of in concert with each other. So it's not just incubation. So we looked at the percentage of visits to the nest together, and we measured this using microchips. So we put microchips in birds, and we had readers on all the nest boxes. So we actually measured this over periods of months, and the birds that visit the nest at the same time, i.e. they both come in a 5-minute period, they're much better at raising offspring than the birds that didn't quite get their act together down here. So essentially, the better they stick together, the better they actually are at producing offspring. And they're also very good at sharing the food. So they both, we can count by videoing them feeding their nestlings. You can actually count the number of regurgitations that a male gives versus a female. And again, I had a student who did literally thousands of hours of this stuff, and males and females actually pretty much put the same amount of investment into their offspring. Most recently, I have another colleague who we looked at sentinel behaviour. So essentially out in the desert, for a zebrafinch, it's a very dangerous place to be. And when a female is incubating the eggs, the male will be within a couple of metres of the nest box, keeping an eye out for predators. And then when they switch over, she'll do the same for him. So again, they're really interested in their partner's fitness, and they're looking out for one another. And that makes sense because you want your partner to stay alive if your partner's well coordinated with you. You don't want to have to go and find a new partner. Another thing that's been happening quite recently is that people have actually discovered that you can measure the personality of all your birds. And literally you could do that for all your own birds, because the more animals that people are looking at with these kind of measures of personality, the more they're finding these quite interesting relationships. So how do you tell the personality of a zebrafinch? You can do a number of different things. So the simplest test is you just release a zebrafinch into a box with 5 perches in it, and you count the number of perches that they land on in the first 5 minutes. Some birds will just sit tight on one perch, and other birds will explore the whole box. They'll visit all the perches, try and escape or whatever. And that's a very repeatable measure of personality. So you can take a bird and do that this week, and if it hits 5 perches this week, that same bird is likely to hit 5 perches the following week. Whereas a bird that sits on 1 perch this week or this month is likely to sit on 1 perch later on. So you can actually measure the personality. And this particular, again, don't worry about the details of the graph, but they literally put a mirror in a cage and looked at how the bird interacted with that mirror. So it's a very, very simple test. You can all do this at home. Some birds will interact with their reflection, other birds will completely ignore it, other birds will kind of try and get away from it. So it might be to do with dominance or whatever, but it's repeatable and it's different. So female, again, in partners in zebrafinches, when they pair females and males up, when a male and female were very close to each other in personality, i.e. when they both were like each other, did the same kind of thing, they were actually much better at producing offspring than a female and a male that didn't match very well. And again, I'm sure we can all think about that maybe in human terms. And then we've repeated that experiment recently in some of the aviaries here at Macquarie, and we found pretty much the same thing, that the birds that are assortative in terms of their personality produce far more chicks than the birds where we've kind of paired them up together with a mismatched partner. So we're really interested in looking at this. Now, initially I thought this was a bit weird about zebrafinches, but actually I've realised that this is actually probably how most Australian birds behave, this very kind of coupley behaviour. And I think there's lots of different reasons why being a good couple and being very compatible is a good thing. I won't go through those now, but again, there's been some work done. So there's a really good group working in North America where they've actually looked across all species of birds to try and understand different patterns of behaviour in things like cooperative breeding and also the strength of partnerships. So this second paper was actually focused on extra pair paternity and divorce rates across all species. So some species have lots of extra pair paternity and lots of divorce, whereas other species like the zebrafinch, they have very low rates of either. And what they've shown in these kind of global analyses, where they've tried to understand these patterns of behaviour by thinking about the climate, is that Australia is actually, Australia and South Africa in particular, are environments which actually seem to kind of foster lots of cooperation, both in terms of cooperative breeding, and we know we've got lots of cooperative breeders like noisy miners and also kookaburras and all sorts of things, but also in terms of low levels of divorce. And the reason for that is probably because of the environment. So the aridity of Australia and the unpredictableness of the habitat means we have very long breeding seasons, lots of opportunistic breeding, so birds can breed throughout the year, and they form very tight partnerships. And I actually think, when you actually think about some of the most commonly held birds in the whole world, a surprising number of them have actually come from Australia, and not just from Australia, but from the arid zone. So actually, I'm kind of starting to wonder, and again, I'd like any feedback from you, whether the reason that some of these birds are really good for keeping in captivity, and they're very easy birds to keep in captivity, is because they tend to form partnerships very easily. And things like cockatoos, if you keep them on their own, they'll form a very tight bond with a human as well. And that's probably because they're just driven by this habitat and our very arid and unpredictable continent to form this kind of pairing behaviour. So one of the big questions we're trying to answer more broadly than just the finches is whether Australian birds are actually special in this way. But then I'm also interested in how that interacts with the whole domestication process, whether our birds are actually more easy to domesticate, and maybe they can be changed by domestication. And maybe when you take them overseas and you keep them in a different environment, maybe the European and North American zebra finches are actually very different now because because of the way they look after them there. Okay, so just as some conclusions, Australian finches provide some excellent opportunities for doing really good science, better than most of the birds elsewhere in the world. But I think what I've learned in the last kind of 3 or 4 years is that the domestication process is likely to be having an influence, and quite a strong influence potentially, on the things that we do. So when we bring birds into captivity, of course we do things that affect the biases, like some birds are much better at breeding than other birds, and then that is going to affect the number of ancestors that those birds produce. So we'd be really, really keen to understand that better, and I think that the study of that, hopefully in collaboration with some of you guys, is going to provide some really exciting opportunities, hopefully for you to learn more about your birds, but also for us to do some science that's otherwise just not possible. So I'd just finally like to acknowledge, so a lot of this work's funded by the Australian Research Council and supported by Macquarie University. The Gouldian Finch work, most of that to date has been funded with the help of Mike Fiddler and the Save the Gouldian Fund, which I'm sure most of you are aware of, if not all. And then the work has been done over a number of years by people who are working or have worked at Macquarie University with me. Sarah Pryke and Leanne Rollins on the Gouldians, and then most of the rest of those people on the zebra finch and the Longtail stuff as well. And then very finally, I'd just like to say that in an effort to try and understand more about the domestication process, myself and Ondi, who's up the back there, and she's come along so you don't get out the door past her, we've prepared a short survey, which hopefully is relatively short. And we'd like you to have a look at this today if you've got time, but otherwise maybe you can take it away and get it back to us, and I can collect them up, or there's a postal address on the front here. This is our first attempt to try and actually see what we can learn from communities like yourself. And some of the questions you might not understand, or we might phrase them a bit poorly, or there might be things that you just don't count. So of course, as scientists, we're interested in counting lots of stuff, and of course I fully understand that you don't necessarily count things in the way that we do, but all information is good. And ideally what we'd like to do is we want to roll this kind of survey out across the whole world of aviculturalists, and you're the first people to kind of give us some feedback and hopefully help to steer the next version of this survey if there's problems with this one. So I'd be really keen if you're able to have a little look through this and either give us feedback tonight So we've got lots of surveys and we've got lots of pens. And as I say, on these kind of up the back. So it'd be great if you could take hopefully 5 to 10 minutes and give us some feedback on this. And of course, I'm happy to take any questions as well. Okay? Thanks.